All right, hello everyone. I hope everyone enjoyed the Laughing Love Bugs. I'm Kira Crass. I am Rare Implementation Co-Lead at NORD in our research department, and I have the pleasure of moderating this next session, How to Participate in Rare Disease Research. And there are many ways that rare disease patients can participate in research and contribute to scientific knowledge about rare disorders. Many patient organizations that I have the pleasure of working with daily drive clinical research forward by maintaining patient registries and biobanks, collecting data for natural history studies, and fundraising and funding research. In this session, we will explore ways people living with rare disease can engage in research efforts to advance treatments and cures. Joining me on stage today, we have Dr. Peng Kong Fox, Science Engagement Director at the CACNA1A Foundation, and Dr. Kevin Glinton, Associate Professor in the Department of Molecular and Human Genetics at Baylor College of Medicine and Texas Children's Hospital. So, welcome, and we can get started with some introductions.
All right, there we go. So good afternoon, everybody. I'm Kevin Glinton. I'm a clinical geneticist and clinical biochemical geneticist, which means I primarily evaluate and treat patients with some kind of genetic disorder. But I also spend a significant portion of my time doing clinical research on rare diseases. So I study a variety of disorders, some of which I've listed here. So Tango 2 related disorders are fairly recently diagnosed or recently described neurologic disorder. So is Mitchell syndrome. A colleague of mine and I have collaborated on describing this new disorder called BPTF-related disorder. And I also study a bunch of metabolic disorders, so GABA metabolism disorders, urea cycle disorders, glycogen storage diseases, et cetera. In my other portion of my professional time, I'm the co-director of our Rare Disease Center of Excellence at Baylor Texas Children's. And so in terms of what this looks like, it's not moving. There we go. How I got involved in rare disease research. I mean, this is sort of baked into the career of a geneticist, right? I'm sure a lot of you appreciate you kind of get diagnosed in a genetics clinic. And as a trainee, this is really what sparked me to do this work. You know, we'd see patients have a diagnosis. And then for me, it was really daunting figuring out what does this diagnosis actually mean for my patient and their family. You know, there's so many disorders now, we just don't know a lot about them, and there's a need for more research on those disorders. And so that's what kind of prompted me to get involved in this. It's become a significant portion of my time. There you go.
Hi, everyone. I'm Peng Kong Fox, and I'm the Science Engagement Director for the CACNA1A Foundation. And I've been in the rare disease space for about a year and a half now. And what brought me into this space is this little guy right here in the pictures with the glasses. That's my son, Alex. He was born in 2015. He's 7.5 right now. And he was my second child. And my first child, Gabriel, who you can see in the pictures too, he's 18 months older. He hit every milestone early. He was sprinting by 13 months. He was singing and speaking in sentences, when he was an early toddler. And everyone said, oh, when you have your second child, they're going to be so ahead of the game as well because they're going to be running after their older brother. And so we had that expectation. And so when Alex was born, he began, everything was sort of normal in the beginning, uneventful birth. He was healthy to begin with. And he's still healthy, but we were starting to see that he was missing those milestones in the first year of his life, but it was never anything so severe that required medical attention. So we just, we kept going on. We're like, even neurotypical children develop at different rates, so it's okay. And then it was finally in the summer of 2017 when his pediatrician finally referred us to our local birth to 3 or early intervention program because Alex was, 15 months at the time and he wasn't walking. He wasn't able to walk. He would take a step and he would just immediately fall over. He wasn't really babbling much. And so, I think that started to be a red flag for his pediatrician. And thus began, the next few years visiting all these specialists that I have listed up here. And they continue to be part of his care team today. But it wasn't until summer of 2021 when we saw a neuromuscular neurologist to rule out his to rule out that his ataxia or his, impairment of balance and coordination, his cognitive delays and his global development delays. We wanted to make sure that wasn't neuromuscular. And the good news was that, the neurologist said, oh, it's not. But then he said, I think there's something genetic. And so he was the first one to say, let's do some genetic testing. And, Alex got his blood drawn in end of, I think it was like right before Thanksgiving of 2021. And by December, we had this diagnosis that he had a mutation in the CACNA1A gene. And this gene is important for neurons to communicate with each other and send information to the brain to the rest of the body. And so that really started to explain why he had these neurodevelopmental, neurological symptoms. So it was a relief, but then you hear it's a rare disease. And being someone with a scientific background, I just said, well, I'm going to learn everything about it, right? Because even though it had been 10 years since I was a scientist, I had got my PhD, I started doing research, I was teaching, and then I stayed home with my kids. I had that opportunity. So I hadn't thought about being a scientist in so long. And so I did my best, and I realized even with that background, it was still hard. And luckily, we actually had a foundation, or that there was a CACNA1A foundation already set up, and it had only been set up, 3 years. And so I was kind of, in some ways, I'm glad that he didn't get the diagnosis until there was a rare disease foundation. But I know for, many of you out there, there isn't that opportunity. And so it does get tough to navigate that. So I'm really fortunate that the CACNA1A Foundation was there. And the minute they found out that I was a parent with a PhD, they latched on. Even though I said, I haven't been a scientist for 10 years, they took the chance to bring me onto their leadership team. And now I'm just honored to be able to help carry out their mission, right? To find these treatments that can help improve the quality of life for our kids and our loved ones. So I'm really thankful for that.
So I think we'll start our conversation with, what is research exactly? So can you tell us a little bit about kind of basic research versus other types of research?
Yeah, so we sort of split them up into these two larger categories. So what we call basic science research and then translational research. And basic science research is really the one I think most of us in the public think of. That's the one where there's actual lab work happening. And that helps us to address kind of what is the actual mechanism of this disease, or how does it happen? How does this change in this one gene lead towards a heart defect or neurologic disorders or seizures? And as I said, this is a lot of times what we actually do in a laboratory setting, because obviously we can't take your child and put them in a cage and study them all day, right? But we can do that for cells that we can grow and harvest and treat under different conditions. We can do that with a mouse or sort of model organisms, where we can put the underlying genetic disorder into a mouse or a rat or a fly and sort of look at what it does to that organism's body, or how that organism responds to different medications, et cetera. And then sort of translational research is where we kind of apply that data to kind of give us idea of how we can better treat this disorder. So if we know that a certain chemical is missing in the body because of a disorder, we can try and see if there's a way to restore that in a child's body. We can explore drug treatments or other kinds of treatments that help give us an idea of how best to treat this disorder. Let's go to the basic framework.
So next up, why should people participate in research?
Yes, so this is, it's a question that is so important, and there's many reasons why, and you might have heard it before, and it may sound like a broken record, but I was just at St. Jude Children's Hospital in Memphis, and I was there to talk about this new initiative that they're starting to take some of their focus and put it on neurological disorders, rare neurological disorders, in addition to rare childhood cancers. And Dr. James Browning, who is the CEO of St. Jude, he was there, and he said, he's looking at all this as saying research before treatment. That's what he told us, right? Because if you're going to treat a disease, if you're going to treat a symptom, right, the best way to do that is to learn what's causing that, right? And that actually goes back down to those tiny micro levels of what's happening in the cell, what's happening inside of the cell. What are these molecules doing? What's the DNA doing? What are proteins doing? And that is really the basis of how we start to develop treatments that are going to improve, the quality of life for everyone out there. And, as Kevin, or as Dr. Glidden, as Kevin was saying, advancing or understanding this disease knowledge, this basic with basic science, that's so important, because the more we know about the disease, the better we can learn to treat what's happening. And again, that supports therapeutic development, right? So what types of treatments are actually going to work for not just a disease, but the separate symptoms within that disease? Because most of us will have not just one symptom, but there's a lot of things that are happening because our cells don't exist in a vacuum. These genes don't exist in a vacuum. They're working in this biological system where one change is actually going to affect hundreds or thousands of other genes. So we have to know what are the right therapeutic approaches that'll work. And obviously this will help accelerate clinical trials as well. That's like the ultimate end game for us here, right? Is that we want to get a treatment into the pipeline and then approved by the FDA so that it's accessible to our communities out there. And we can't do that without that basic disease knowledge and then learning how to develop those therapeutics. And like I said, I keep going back to, our goal is to improve the quality of life for your community. I've been in the rare space for, a year and a half. I've been with the team at the CACNA1A Foundation for just under a year. And I'm so uncomfortable using the word cure, but I don't think it's a bad thing, right? Because I think cure means something different to everyone. And so for me and for our team at the foundation, we often talk about, what is actually needed, what's important to our patients and their families, right? That cure could just be a 10% improvement in their mobility, or that they're able to say mommy or daddy, right? And that's enough for now. But that doesn't mean we stop there, right? We're still going to be moving towards treatments that are going to have an even larger impact, but it's not going to happen right away. And that's where we have to keep research going, because we know that diseases progress and they're going to change over time and the treatments are going to change. And the only way we're going to learn about this is by talking to all of you out there, because you guys hold the most valuable information that no scientist, no researcher, they're not going to know, right? It all comes from you. So this is, you're raising awareness, but not even to the general public, right? You're raising awareness to the people who are actively playing a role in getting these treatments to the pipeline. You're showing researchers that you care. You are empowered to participate in research that's going to bring those treatments. You're going to, you're raising awareness to the FDA that, again, you care enough so that they know how important this is to you. You're raising awareness to the industry, the companies out there. They're going to see this disease has 200 patients waiting, lined up to enroll in clinical trials. That will make an impact. So again, lots of reasons why. These are just a few.
So speaking of clinical trials, why don't we talk a little bit about the pathway to clinical trials?
Sure. So this is a slide that I've used in some other talks as well, and it's just a really simplified version of how we at the foundation, I think other foundations as well, are sort of paving our pathway to clinical trials. How do you get from a rare disease all the way to moving something into the pipeline? And there's a lot of steps here, but with just everything that we've been talking about, it kind of shows you where the basic research comes into play, right? That first purple box, you're building your preclinical toolbox, building all the tools that you're going to need to move things and treatments into the pipeline, and learning about those disease mechanisms. And then once you get past that, you have your translational research. You're working with people and institutions to build those therapies, to figure out which ones are going to work, and then it leads to what we call these proof of concept studies. And these studies are so vital because these are the studies that show the FDA that whatever treatment you're testing works in your disease. Even if it's just in a cell or a mouse, that is evidence that shows this could work in you. And that's so important. And so, anything that happens before we get to the clinical trial pipeline, we call all that just preclinical research, right? So you hear all these things and you're like, well, what does it all mean? It's all that work that needs to be done before you get to the clinical trial pipeline.
And I was going to say, I'd like to point out, every rare disease is at a different stage of its sort of maturity as an understood entity. So there's some rare disorders where they've only been described for about a year or two. And so those are going to be primarily involved in the preclinical toolbox phase. There's other more mature disorders, like we heard about SMA this morning, that's actually really further along in the pipeline, but there's still clinical research that still needs to be done for sort of long-term proof of concept studies, things like that, or efficacy. So it just will depend on, your particular disorder where, your group fits in terms of fitting into the clinical trial pathway.
And I just want to add, too, that this isn't a linear pathway, right? I've drawn it as one, but I think you all know, right? It's sort of like 2 steps forward and then like 25 steps that way and then 25 steps that way, right? You're going to have times when you need to pivot or you need to go back and look at something else. I mean, that's how science works, right? So don't be afraid when something doesn't work out right and you have to go back, because ultimately that's going to get you forward.
Yeah.
Absolutely. So we've talked a little bit about the importance of research, why you should do it, but how exactly can you participate in research? What can we do to participate?
Yeah, and so yeah. But I think so. Like, once again, this goes back to the concept of where your particular rare disorder is in terms of its maturity, right? So for some disorders that are really, really rare and really newly described, you're going to have to start just with what is known about it, like how many papers have been published, how many patients are there. Peng-Kong and I work with rare disorders where there are less than, several dozen patients. And so you kind of have to start with what's published in the medical literature. And for most doctors, we use this resource called PubMed. This is an NIH-funded sort of compendium of medical research, where you can kind of put in, the particular gene that that is deficient in your rare disorder, or the particular disorder itself, and see what's out there, what's known, or who are the people publishing about this disorder. You can also honestly just use regular old Google. It's still great. A lot of us doctors still just will start with a Google search first, either by the disease or by the disorder name itself, or just starting from there. And then I would always say as well, start with that doctor who gives you the diagnosis. Like, well, what did you read about it? Who's looking at this? Who's researching? Have you reached out to that group in France or Italy, et cetera, who's described this? But ask your doctor to really kind of explore what they know and, what they've kind of reached out and sort of gathered from the literature itself.
And just going on what Kevin was saying there too, when you find those research papers, I know that they're really hard to read, right? I mean, I have a science background and I still have to look up words and reread papers over and over, and sometimes I just skip to the discussion, right? Just get to the point. But there are always, you look at the authors, you look at who has written these papers and where are they located, and don't be afraid to just give them a cold call or a cold email. Because a lot of times these researchers who are studying rare diseases, they've never met a patient, right? They've studied it in the lab, they study it in cells and maybe some animal models, but it makes such a big impact the first time they meet a patient with this disease. And sometimes that gives them the drive to actually say, oh, this is real. This is not just in a cell. This is the real thing. And so don't be afraid to email them again, because they're busy. There's a lot of things going on. If they didn't email you back the first time, it's probably because they saw it and they said, oh yeah, get in line after those 25 emails that I have to answer. So don't be afraid to reach out again. And then build your network as well. For those of you who maybe are interested in starting a patient advocacy group or an organization, it's all about networking too. I mean, I know that NORD has resources about how to start one, but I would say talk to other PAGs. I mean, that's how the Kaplan-Meier Foundation also got to where we are, is that you talk to other PAGs who have been around, and so many of them will offer you resources. They don't want you to reinvent the wheel, right? If they've already done the work and they can help you, I've never had anybody that we've spoken to who said, no, we're not going to share that with you. So take advantage of that, and again, take advantage of the resources that NORD can provide, and go to conferences, right? If you're able to, if you have the resources, go to conferences that maybe are in the area of your disease, or ask your doctor what conferences they go to. This is where I think it really comes down to networking and putting yourself out there and meeting as many people as you can.
I believe the next slide is also a little bit more about preclinical toolbox.
Yeah, so one of the ways that you can participate in research, and this is something that we really have been encouraging our families to do as well, is to help us build our preclinical toolbox, right? Because that basic science is needed before you can find those treatments. Just like St. Jude says, research before treatment. And so one of the most important things that you can support, right, is what we call a natural history study. And this gives you a full picture of what your disease is over time. It can answer things like how many patients there are, how many variants are those mutations, how many changes are there in the gene. You can start to think about, the correlations between your variants and your symptoms. You might hear it referred to as genotype-phenotype correlation, but it's basically how does this variant that you have lead to the symptoms that you're exhibiting? Again, what are the symptoms? How do they change over time? When do they start? Any current treatments you're on, have they changed? All of this is so valuable because, again, it's something that nobody else knows except for the patient community.
And yeah, I was just going to add, I think we tend to overlook the value of natural history studies, but they really are the best way, as Peng-Kong said, to get a full picture of this disease. And honestly, that's going to be the most important thing for you as a patient, right? When you first get a diagnosis, like, what does this mean for my child or for myself? How much life am I going to have? What is my life going to look like moving forward? And a lot of these questions we can't answer very easily for rare diseases because they are rare and patients can look differently, and it's hard for us to kind of extrapolate from one patient to the next how things are going to look. And so we build a lot of these natural history studies in our department and to really get the patients together and look at things from a macro level, like, well, how many mutations do we see? What does that mean for patients, et cetera? And we have found, just anecdotally, right, for one of the disorders I study, Tango 2, that when we talk to the patients, we learned that some of the patients were taking a B vitamin supplement, and that actually seemed to prevent them from having episodes that sent them to the hospital. And we would not have known that if we hadn't talked to all of the patients and sort of, gotten, history of what medications they were on and kind of traced out like what the effect was for each one. And so that's what can be really important. You as families, you do lots of things for your children and your doctors help you do lots of things. But for rare disorders, it's hard to know if those things work because we're just kind of looking at you as a isolated patient. And so when we can put them together as a group, it does have so much value that I can't really emphasize enough.
Yeah, and I also just want to add to that too, is that something that's really important that comes out of these natural history studies is it helps to design effective clinical trials, right? It tells you what you should measure, how long you should measure, maybe what type of change you're going to be looking at. And a lot of times clinical trials might fail because you don't have the right baseline measurements or you don't actually know what to look for. And so we can get that information, again, from all of you out there. If you go, if you participate in these natural history studies, it gives us that basic information to design the really, again, the most effective clinical trials to clear the way for, FDA approval of potential treatments. And then another thing that is also important in building your preclinical toolbox are these research tools. And Kevin kind of alluded to it earlier as well, is animal disease models. We don't want to study these in humans because it's not ethical, right? We can't keep people in cages, and it takes a long time, right? But we can use these models, these different systems like bacteria or cells or mice or flies or zebrafish. They have the potential for us to go in, or for us, but for researchers to go in and genetically manipulate those genes and give them these pathogenic or disease-causing variants and see what happens, right? And hopefully, if, in a good case, it's going to mimic exactly what happens in the patient population, and you've got the perfect model to study how to treat this disease without having to even touch a human being yet. But something that can come directly from patients and directly from all of you are what we call these patient-derived cell lines, iPSCs. And really, okay, so induced pluripotent stem cells, what does that mean, right? These are cells that have the ability to turn into any other type of cell. And when they come directly from a patient that has a pathogenic or disease-causing variant, now you've got a cell that, again, has the genetic, the genetic background that the patient has. And you can differentiate them. In our case, we want to look at neurons. So we can differentiate these iPSCs into neurons and start adding a drug and see what happens, right? And so again, it gives us the perfect environment in which to study the disease outside of a human.
Do you want to talk a little bit about specifically the CACNA1A Foundation's clinical?
Sure. So I won't spend too much time on this. I know there's a lot of information here, but I just wanted to give you an example of how we at the CACNA1A Foundation have been building our preclinical toolbox for the last 3 years, and it's still an ongoing process. And this is not a pathway that everybody has to take. There's a lot of different platforms that you can use to build natural history studies. That first column, we actually do have it's a virtual natural history study that's being run by Dr. Wendy Chung at Columbia University. And again, it's there to really learn about the progression of the disease, and it's patient and caregiver reported data. And the target audience is really for academia. It's also, once it's published, it's available for anyone to see and learn about the disease. We have a data collection program, a study being powered by RareX, another entity that is helping us collect data. And this data platform is actually specifically for data collection of rare diseases. And again, it's patient and caregiver reported data, but it gets our data into the eyes and the hands of other people that can be outside of academia. And then I really just want to point out the Citizen platform that's by Invitae, which is one of the big genetic testing companies out there. This is a different kind of natural history study where patients can go in and essentially collect all of their medical records in one place. And then data scientists can look at your history, and they can basically construct a retrospective natural history study. How did this disease progress, like the moment your first symptom showed up? And that's really important too, because industry or pharma companies want that high-level data. They want that data that's validated by clinicians and providers. And then, we also have our biobanking program with our patient-derived iPSCs, our animal models as well, and then something called biomarkers, which is, again, those really important endpoints or things to look for and measure when it comes to those clinical trials. So if anybody has any questions about how we set these up, please come and talk to me. Like I said, you don't have to have everything in here. Please don't. It's very, if you have the capacity, great, because we're a large gene. We have a lot of mutations. We sort of want to have a way to collect as much data as possible, but it can be overwhelming on the patient population. So find what works for you. Think about what you want to use that data for, and go from there.
Yeah, and I was going to say, the CACNA1A Foundation is obviously, a very mature and well-organized organization. But don't feel like you have to jump to this stage immediately, right? It is daunting sometimes to realize how much research can happen from a disorder. But, if you just organize as a group and get started, I think that's just one step that you can move towards this stage. Also, finding a collaborator in academia can be super helpful because they can help you sort of navigate these relationships between a laboratory or a pharmaceutical company. So Dr. Wendy Chung is an amazing investigator, and she has a lot of experience with this. And so finding someone who is studying the disorder and getting their advice or adding them to your scientific advisory team for your disorders foundation can be super helpful to kind of move things along this pathway.
And I just want to slip in that the foundation's actually only 3 years old.
Right?
No. But that's the thing is that we were
This
able.
is possible.
Well, no, but we were able to do it because of networking, because of finding the right people to help us, and talking to other PAGs, again, who said, hey, you should try this, you should try this. It is a lot of work. it can be done if you have a good team too, which we're really fortunate to have. So it can be done.
Definitely.
I think we just have one more thing we're going to say before we move to some questions, which is a little bit about enrolling in clinical trials. Maybe that'll be our final topic before we open it up for questions.
What do you want to say about it?
How to enroll in clinical trials, you mean? Yeah, so there's lots of different ways to kind of go about this. And once again, this gets to, at what state your disease is in terms of its progression along the pathway. So for some newer disorders, it may just be reaching out to who is studying that disorder and figuring out if they have a natural history study going on. Or it may be if your disorder is further along down the pathway, reaching out to the drug companies or going to clinicaltrials.gov to see if there's a trial open for your disorder and what state it's in. Some of them may just have natural history studies that are run nationally. Others may have sort of drug trials already going and enrolling that you may be able to contact the research coordinator and be a part of. So it will just depend where things are. Hang on.
Yeah, I would add to that too. Reach out to any patient advocacy groups that are established for your foundation or for your disease, because we are actively working to get to clinical trials. We're identifying clinicians who could help lead these trials. We're doing these natural history studies to find the best ways to design these clinical trials. And so the minute that we know that there's something started, we will put it out there to our community and make sure that everyone who's eligible has the opportunity to learn, or enroll.
All right, great. Thank you. I think we're going to open it up to some audience questions.
We do have some in. First question is, can you explain exactly what a biobank is and how to participate in one?
Great question. So as Peng Kong was saying, these biobanks are places that sort of store your genetic data or some of your biological data. So it may be that they collect a skin biopsy and then grow up stem cells from that and keep them in a freezer. They may collect blood samples or buccal or cheek swabs, something where they can grow up sort of your DNA or your cells and keep them available for doing further experiments with. And there's various different avenues for participating in a biobank. Some of them are sort of centralized for different disorders. Some of them are disease specific. So it just will depend on the particular condition. But
Yeah, so our biobanking program is essentially where we collect blood samples, and so I should start over. We're working with Combined Brain, which is a consortium for rare neurodevelopmental diseases, and it's up to about 60 different organizations right now. But they have really opened the avenue to provide places like a biobanking program where we can collect samples from patients, have them turned into these iPSCs, and then store them for us. And then if somebody comes along and says, "Hey, I want to study this," they can send out those lines. They can send out those cells. And so that's how we've set up our biobanking program. But as Kevin was saying, it can be for any patient-derived model system as well.
it's important to note these are very well regulated. That's something patients ask us a lot, like, well, who's going to have access to my cells or my data, et cetera. These things are very well regulated. Usually you're asked to sign a consent form where they go through the regulations of who has access to everything. So, but it's important to read that information before you decide to participate.
Okay, how can a patient find out if there is an ongoing natural history study for their disease?
So I would say, again, if there is a patient advocacy group out there that represents your disease, reach out to them, because there's a good chance that they already have one or they're about to launch one, and that's going to be the best way in. If there isn't one and you're interested, then Google your disease, right? Or go to PubMed and put your gene name in, find some clinicians and some researchers and reach out to them as well, because there's a chance, it might be a small chance, but if you look hard enough, you might be able to find somebody who is, or who knows somebody who's doing a natural history study for that gene. It's all about, you do have to take the initiative a little bit if there isn't a patient advocacy group, but there are resources out there to help you with that.
And I will say, so when you go to literature, there's always usually a paper that the title is describing the first time this disease is described, or like this diagnosis in a certain amount of patients, like 12 patients with Tango 2 disorder. And so I would recommend looking for your disease, but those kinds of papers where they look at a group of patients or a cohort describing what happens in your disorder. And usually the person who is the first or last name in the list of authors is usually the one who collaborated and got all the patients together and got all their information. And that's usually, not always, but usually the person who's interested in doing an ongoing natural history study. So I personally have been emailed a lot by some patients because I wrote a paper a couple years ago for our BPTF-related disorder. And we got a lot of these emails, and I answer all of them because we are collecting patients looking to do a natural history study. So it is possible, and it does happen, but it does sometimes take a little bit of initiative, because for busy clinicians and researchers, it's hard to kind of go out and advertise for a lot of these things.
Next question is, who is the best person to consult with about whether I should enroll my child in a clinical trial? For Dr. Glinton, how do you counsel patients on participating in research? And Dr. Fox, does your foundation counsel patients and families about participating in research?
Great question. So who's the best person? I think, it's always important if the affected person's a child, right, to have a discussion with your family doctor about, just getting a good sense, because that's usually the person that you trust the most in the medical community, right? But also having a discussion amongst yourselves and the family, like, is this something we're interested in? What is the trial? Do we want to go through this and get our hopes up, or maybe very little outcome per se? But I think the other part of the question for myself, when I discuss a trial with patients, there's lots of different factors that I consider, right? What are the risks and benefits of participation in a trial? For natural history studies, I think there's very few risks that we talk about. The major one is just the loss of sort of privacy of your information, but we mitigate that pretty easily. For other trials, it is more of a discussion, because for certain treatments that are coming on board now, it can be sort of little bit trickier. How stable is your child, or is the patient? How much do we think that they may benefit from a treatment if it's going to cause side effects somewhere down the line? So it is an open discussion between the referring geneticist, myself, and the patient about how much you think this is going to be useful for you and your family versus how much you really want to participate and move the field forward.
Yeah, I echo that. You should talk to your child's or your primary care provider, your doctor, because they will have the ability to sort of gauge whether, based on your conditions, based on your symptoms, they can help you decide, the risks and the benefits there. But as a foundation, we're not there yet where we have to really start talking about it. Hopefully we will be in the next couple of years, but we always just try to educate everybody as much as possible. What is a clinical trial? What are the risks and the benefits? What are the different stages? What should you expect? We really try to emphasize and educate our community. And it's going to differ for each clinical trial too, right? It's not that everyone's going to be eligible for all clinical trials. There could be a clinical trial that we know is actually really going to, or the best way to test it would be to test it on a small cohort of patients with certain symptoms. So we're going to, target them and educate them about what that is. And the same for, if there's another trial that's set up to improve ataxia or balance and coordination, that's going to be a different set of patients versus those with seizures or epilepsies. So, so I think the best thing to do is to, again, talk to your primary care provider, talk to your child's or your specialist, but please consult too with your, I shouldn't say consult, but you can always reach out to your patient advocacy group to be educated on, the other aspects of clinical trials as well.
We have a question for Kira. How can I participate in a research study within NORD?
Sure, hello. So there may be a registry on our platform in your rare disease, and where you would look to find that would be on iamrare.org. We have a link on the front page that says find a study, so you can look by your disease, and there may be one that is in your rare disease. If there is not, and you're part of a patient advocacy group and you're interested in starting a registry, you can contact us at research@rarediseases.org, and someone can get back to you to talk to you a little bit about what is involved in starting a registry and gathering information on your particular rare disease.
Thank you. That's it for questions.